The overall goal of this procedure is to directly quantify red blood cell aggregates in microcirculation dynamically under controlled and constant shear rates. This is accomplished by first entraining red blood cell suspensions in a double Y microchannel with a phosphate buffer saline solution, creating a constant shear in the blood layer. The second step is to visualize and record the flow with a high-speed camera. Next, use image processing techniques to process the recording and obtain an average aggregate size and the aggregate size distribution in the microchannel. The final step is to determine the blood velocity by following the displacement of fluorescent particles injected in the microchannel between two consecutive frames. Ultimately, the shear rate in the microchannel is calculated based on the blood velocity and the blood layer thickness to relate the aggregate sizes to the corresponding local shear rate.
The main advantage of this technique over existing methods, like the light transmission method, is that the red blood cell aggregates are analyzed dynamically in a microscopic shear flow, as opposed to statistically with other techniques. This method can help answer key questions in the hemorrheological field, such as understanding the conditions of red blood cell aggregates formation and defining blood rheological behavior in microcirculation.
To begin, fabricate microchannels 120 microns wide, 60 microns high, and 7 millimeters in length according to standard photolithographic methods. Next,